Good afternoon, everyone. My name is Amar, Pegu. I work at the Vaccine Research Center under John Muscula's group. And today I'm going to share some data from our non-human primate studies using bispecifics and other engineered bNAbs that's done. So as Nancy just pointed out, there are several proof of concept studies in non-human primates that have shown bNAbs can prevent SHIV infection, and the AM trial is about to address the question is, can an antibody actually provide protection against HIV infection, because that hasn't been shown so far. And at the VRC, we are involved in developing next generation bNAbs that can be used for prevention, that have longer half-lives, increased potency, or greater breadth. What we also understand is that no single bNAb would be able to protect or provide protection, since they don't neutralize all the circulating strains. There's about at high potency, and targeting multiple sites would be required. So a cocktail of 2 or more antibodies like the 3BNC117 with 101074 or other combinations would be required. And therefore, there's a lot of next generation antibody technology that has been used in industry now that have developed antibodies that can have multiple specificities in the same molecule that will potentially lower the cost of manufacturing, delivery, and administration. So in that regard, we started work, this paper we published in 2015 looking at bispecific antibodies, in which we had made VRC07 and PG916 like bispecific, which had great potency and breadth. And then David Ho's group also has now a clinical product that's in clinical testing, the 10E8 with the anti-CD4 antibody or bispecific in this format. And then Michel Sunzak's group also had IgG3 type of bispecific in which he combined these 2 antibodies, and it also showed good potency and breadth that was about 90%. So in collaboration with Sanofi, we came up with even a newer format, which is the trispecific antibody format here, where we have one arm, which is a single arm, has a regular FAB from just a VRC01 in this case. And then in this, on the other arm of the antibody, we put the 2 variable regions in conjunction to each other, linked via various linkers. And this was using their technology called crossover dual variable immunoglobulin domain. So you can put about 3 different antibody specificities in one molecule. And we've shown that this is how the structural model looks like. We have VRC01 on one arm, and PGDM and 10E8. They can all bind to their respective antigens. And then when we looked at the breadth and potency, these antibodies now are one of the most potent and also very broad. So you can see some of the more potent antibodies here have breadths that are really lower than 80%, whereas some of the more potent antibodies don't have very high breadths or potency. So these bNAbs are now equivalent to some of these better, has the properties of the potency and the breadth for the bNAbs. And then using the non-human primate model, what we wanted to test was, do these trispecific antibodies behave like antibodies? And in terms of PK, they look very similar to a regular VRC01-like antibody. And the N6 trispecific containing also has a PK comparable to the N6. So they do have kinetic PK properties similar to antibodies. And when we did a mixed challenge experiment in non-human primates using viruses, a cocktail of viruses that were resistant to VRC01 or PGDM 1400, and did a passive transfer using the trispecific or the parental antibodies, we can see breakthrough viruses or breakthrough of resistant viruses in the VRC01 group or the PGDM 1400 group. But in the case of the trispecific, you can get protection against both the viruses that we use, suggesting that we did also have in vivo proof of concept that they can protect in this model. So going forward on that, we also now wanted to look at immunotherapy. Does this antibody also have an antiviral effect? For this, we used the SHIV ABG505 that was developed by George Shaw's group. And in this, we treated with the N6 trispecific, which has a very good potency against this virus, at week 6 and 7 post challenge using a 20 mg per kg IV dose. And we used 6 animals in this case as a proof of concept, where we see the viremia just before infusion. There's a wide range of viremia here, but when you give the antibody, you see a very potent dose, about 1,000-fold decrease in viremia, suggesting a very strong antiviral effect. And then there's rebound, which correlates very well with the antibody levels, which is the black lines here, and the red ones are the viremia. This is 6 animals shown individually. You can see as soon as the antibody level drops below about a threshold of about 10 micrograms per mil, you see the rebound coming up of the virus, suggesting that as long as you maintain expression or the protein levels and the serum about 10 micrograms per mil, you get good suppression. In In terms of long-term viremia, these are the control animals that were not treated. You get like half of the animals start controlling. Some of the SHIVs are controlling, as has been mentioned. And in the trispecific groups, there is more control, but this is a small scale study. This is not meant to look at long-term control. So we are following up that by increasing animal numbers. But overall, what we can see that we've used this model to develop the trispecific antibodies that can provide protection to NHPs in a passive prophylaxis method. And then also we can also look at the antiviral effect where we see a greater than 1,000-fold decrease in viremia. And we are doing ongoing efforts to assess long-term impact of these trispecific bNAbs. And based on these data, we are Sanofi, and we are also started the clinical trials of one of these trispecific antibodies in humans. In terms of HIV treatment and cure, as all of you are aware, there's a latent reservoir of HIV-1 infected cells that are not eliminated by antiretroviral drugs. And antibodies alone cannot target this latent reservoir because there's very low levels or no expression of envelope on the surface of these latently infected cells. So how do you target this? As we are all aware, there's a kick and kill or strategy that is known to propose activate and eliminate latent reservoir. In that regard, we have published previously in 2015 about a novel multifunctional antibody, or which is called a T-cell engager, in which you have in one arm of VRC07 with an anti-CD3 arm, which can both activate and lyse latent cells. So this is a concept that is not very new. It has been used in targeting tumors. As you can see, the bispecific antibody can bring a CD3 expressing T cell close to a tumor target cell and then cause the lysis. And then additionally, in cancer, there's a very hot area of research on the checkpoint inhibitors like PD-1, PD-L1, CTLA-4 to increase this activity of killing of these T cells. So can we see something in HIV or SIV? In the case of HIV infection, it has been now widely reported that due to the persistent nature of the infection or chronic infection, you get effector CD8 T cells activated. But over a course of period of time, there's chronic activation, which leads to exhaustion, which is marked by high expression of some of these markers of exhaustion. So using the SHIV model, can we also model that in our SHIV-infected animals? So when we looked at viremic SHIV animals and looked at the T cell subsets in lymph nodes, we can see that the PD-1 is really highly overexpressed in the SHIV-positive lymph nodes compared to SHIV-negative. And then, in fact, in the terms of the TIGIT, which is another checkpoint inhibitor, you see much higher expression of these markers compared to non-infected animals, suggesting that this model might be a good way to use these therapies to see if they have any efficacious measure. So in that context, we've also engineered now trifunctional antibodies. This was the original bifunctional antibody that we published in 2015. So we've now made trifunctional antibodies. This is separate from the Sanofi trispecific, in which we just added an anti-TIGIT or anti-PD-1 targeting arm to these bifunctionals so that they can target both CD3 and anti-TIGIT, anti-PD-1. And then, so using an autologous ex vivo system in which we use CD8s and CD4s from SHIV-infected lymph nodes, which have high expression of TIGIT and PD-1, we can show that when you incubate them, co-culture this regular CD4s and CD8s in the presence of these different antibodies, you can see that this is the original bispecific, the VRC07 anti-CD3. You get some reduction in the GAG DNA in this. We have similar data for RNA also. We get some decrease in the GAG DNA, but when you do with the TIGIT or the PD-1, the blue and the green, you get a much greater decrease in the amount of the CD4 cells that have the GAG DNA or the RNA levels, suggesting that we are making these effector cells much active and better, or we are getting over the checkpoint blockage that they have in terms of effector activity. So to test this formally, we have now started some pilot animal studies. This is just 3 animals per group. So we have animals that were injected with VRC07 anti-CD3 anti-TIGIT at 100 micrograms per kg versus an antibody that has the bispecific, but not the TIGIT arm is changed to an anti-RSV. So this is just a bifunctional, just VRC07 anti-CD3 to see if there's a difference in the effector activity and what happens to the whole blood cell count, and also looking at the killing activity. So in terms of the results, what we found is, first of all, we looked at just whole blood cells and looked at what happens to the cells. We have published this previously that with the bifunctional antibody, you get rapid depletion or migration of the CD3, CD4, CD8 cells from the periphery to other sites. And we do see that by 6 hours, you get a drop, and then that's maintained to day 2. In the case of the TIGIT antibody, we also got similar because it also has the anti-CD3 part here. So that was not surprising. What we wanted to look more closely was that it looked at lymph node T cell activation. So at day 2, we necropsied the animals and looked at what happens at the lymph node. And here what we found was, in the case of total CD4s and CD8 T cells, we can see there's activation as measured by CD69 positive and Ki67 positive cells, double positive cells. You can see post-TCE infusion, you get much higher activation. And especially in the case of the CD8 T cells, you get much greater activation with the TIGIT trifunctional compared to just the bifunctional VRC07 anti-CD3. And when we looked closer and which subsets of CD8s are getting activated, you can clearly see that it's basically the follicular CD8s or the non-follicular CD effector populations and the central memory CD8 T cells, that all 3 animals you get some degree of activation of these cells. So these cells are not getting activated. Are they also having better killing activity? So for that, we did an ex vivo type assay in which we sorted CD8 T cells from the same lymph nodes from these same animals and used a target CM HIV-infected cells as the target cells to look at killing, and just incubated them at 12 hours and looked at how many cells are getting lyased from these. So what we found was that when you have the TIGIT compared to the RSV, you get much better killing after the TC infusion compared to just the bifunctional. So these cells should also have the bifunctional. They should have some killing activity, but with adding the TIGIT arm, you really enhance the killing activity in this case at day 2 post-infusion. So this suggests that using this intervention or targeting the TIGIT molecule, you can enhance or make these CD8 T cells recognize and kill infected cells much better, suggesting that there's a therapeutic benefit here. And we've tried this in vitro using combinations of these antibodies. It's always better when it's on the same molecule rather than having a combination of Fab and a bifunctional antibody. So it suggests that you need to have it on the same molecule to target these cells. So overall, using the non-human primate model, we can actually address some of the chronic viral infection CD8 exhaustion models that we have seen in humans. And then the single infusion of these T-cell engagers are safely tolerated. We are now trying the same with the PD-1 trifunctional antibody. We see redistribution activation of immune cells. And then we do see improved cytolytic activity. We are doing more imaging studies to look at what happens to the virus. Is it going to the follicles or not? And then we are doing ongoing animal studies to assess actually the efficacy of these T-cell engagers. Finally, I would like to acknowledge all the people involved in this research, John and Rick, for all the guidance. One Lisa was leading the T-cell engager project, the TRP, George Shaw for providing the virus, Jeff and the NCI colleagues for all the sequencing and the virus load, and Sanofi for providing us the trispecific antibody. Thank you. I'll take any questions.
All right. Go ahead. Jeff Liston, Frederick National Lab. Amar, nice talk. I always wonder, and maybe you can fill me in. Rosie's talk was interesting because using native rhesus-derived rhesus antibodies in rhesus simplified things, didn't see any ADA and so on. Can you comment with some of these more exotic, unnatural forms of antibody-based reagents that have alternative structures and configurations, conformations, about overall immunogenicity propensity for development of ADA?
Every time you give a human IgG, there's always an ADA response, and the trispecific or the trifunctional, you also get an ADA. So we don't see there is an increased level of ADA. So that's why I think studies with Mario's antibodies to make trispecifics with those SIV native antibodies and also trifunctional would be the best way to address the question. Is it really the format, or is it just the human versus?
Or the best way will be your clinical data.
And the clinical data, obviously. Yeah. But if you want to model it in anonymous time.
And second question, with regard to your combined antibodies that include a CD3, anti-CD3 moiety, I've heard other people pursuing that approach talk about basically trying to modulate the avidity of the CD3 binding activity of that to try and titrate immune activating potential and produce the desired effect without causing a generalized immune activation. Can you comment on that just in general and how you're approaching that?
Yes. So we have tried different versions of CD3 that have different affinities for the CD3 and found that there's you can get very high binding and affinity that can lead to much more toxicity. So we have to damp down on the affinity to have properly modulate that. Yeah, that is very important.